Hey guys, today we're doing chapter 9, microbial nutrition and growth. So what is nutrition when it comes to microbes? It's the process by which nutrients are acquired from an environment and are used in the cellular activity such as metabolism and growth. Facts about cellular nutrition. First, an essential nutrient cannot be produced by the organism, and an essential nutrient must be obtained from the environment. The acronym for essential nutrients is the CHNOPS, which is the carbon, the hydrogen, oxygen, nitrogen, phosphorus, and sulfur. The CHONPS, CHNOPS. Our first one is our C, which is carbon, and there are two types of carbon sources. Okay, there are two types of microbial carbon sources. One is the heterotrophs and the autotrophs. And the heterotrophs use photosynthesis to derive organic compounds from carbon dioxide. And a heterotroph example is animals, fungi, different things like that. The autotrophs, they use inorganic carbon dioxide as its carbon source. And an autotroph example is algae, different kinds of plants, those kind of things. And I did list some examples on this slide. Our H is hydrogen. Hydrogen helps maintain the pH of cells. It does assist and help form hydrogen bonds, and it is a source of free energy. Our N, which is nitrogen, very quickly, it is the most common gas in Earth's atmosphere. It does make up 79% of that. Our O, which stands for oxygen, oxygen makes up 20% of the Earth's gases. And it's a major component of lipids, carbs, proteins, and nucleic acids. Talking about our P, which is phosphorus. Organic sources of phosphorus are listed below: DNA, ATP, RNA. There's a couple of different ones. The main inorganic source of phosphorus is phosphate. Our S, which is sulfur. It's found within rocks and other sediments. Sulfur's unique linkages, it determines the shape and the stability of proteins. There are two types of essential nutrients, the macronutrients and the micronutrients. Of course, macro means large, micro means small, but it is really important that we remember that sizing is referred to the quantity that is needed, not an actual size of the nutrient or the microorganism. It's just the quantity of nutrient that is needed. So micro meaning small is small trace elements such as zinc or nickel. Those were just a couple of examples. And on this picture down here on the left hand side of the screen is a picture of zinc, which is one of the micronutrients. The other one is macro, and these are nutrients that are needed in larger quantities. What is a growth factor when it comes to microorganisms? A growth factor is an organic compound such as a vitamin or amino acid that must be provided in the diet to facilitate growth, and it's an essential nutrient. This picture that I have over here to the left-hand side is a picture of an amino acid. Feeding microbes, there are two different ones that we need to remember. Phototroph and chemotroph. And phototroph is energy is from the light, photo as in photosynthesis. Chemotroph is they gain their energy from chemical compounds, so chemical. So we're going to talk about diffusion real quick. So I listed the actual definition of diffusion on this slide, and there is an example of it down at the bottom. but I'm going to put it in a little bit more simpler layman's terms that's easier to understand. So let's say that I have a glass full of water, um, and I have a red Kool-Aid packet, and I'm going to make Kool-Aid. So my my larger molecules are the water. So when I add the Kool-Aid packet, those molecules are not going to just um, sink to the bottom and not have any red traces, or they're not going to just stay on float on top of the water. They're not going to go in straight lines or anything like that. They're going to diffuse all through that water, making the whole water red. So I hope that's a little bit more easier to understand. Okay, on to osmosis. It is the diffusion of water across a selectively permeable membrane in the direction of a lower water concentration. So again, if you have a thing full of water, think of it as like you have a sponge, like you have a square thing of water, and you have a sponge right in the middle of it. The sponge will act as like your selectively permeable membrane. So some of this water will pass through into the lower water concentration areas, which may be on this side. This side might have more salt or different solvents in it. So it passes, it's the diffusion of water across the selective permeable membrane in the direction of a lower water concentration. Okay, guys, in regards to osmosis, it depends on the environment and the concentration of the solutions with the cytoplasmic membrane. So these are a couple of different environments and things that can affect osmosis, and these are some conditions. First, we've got hypertonic, which is outside of the environment. It's a slightly higher concentration of solutes than inside the environment. Then we've got hypotonic. It's a concentration of the outside environment is lower than the cells of the internal environment. And then we've got isotonic, which is the most stable environment for cells. Now, hyper meaning high, we can always remember that is the higher concentration. Hypo meaning low is the lower concentration. And then isotonic is the most stable. Right now we're going to talk about some environmental influences on growth. One of the big ones is temperature. We can have a minimum temperature, a maximum temperature, and an optimum temperature. The minimum, of course, is the lowest temperature that an organism will grow. Maximum is the highest temperature at which an organism will grow. And then the optimum is the temperature that species show the most rapid growth rate. This is what they thrive in. The psychrophile, which is cold, it likes the temperature at low. It likes the icy temperatures, 0 degrees Celsius to 20 degrees Celsius, with its optimum temperature is 0 degrees Celsius to 15 degrees Celsius. So the psychrophile, it does prefer the cold. Then going to the maximum, the thermophile, it likes the heat. They thrive at a temperature of 50 degrees Celsius or higher. So these are more like our Florida microorganisms. And then our psychrophiles are more like our Alaskan ones. Optimum is the temperature at which they grow their best. And the mesophiles are microorganisms at intermediate temperatures that grow best at intermediate temperatures. So they may be somewhere in between the minimum and the maximum. But anyway, we can look at the psychrophiles as cold, the thermophiles as hot, and the mesophils as they're just right. Microbes fall into one of three categories. Number one, those that can use oxygen and can detoxify it. Number two, those that can neither use oxygen or can detoxify it, or 3, those that do not use oxygen but can detoxify it. We're going to talk about oxygen requirements really fast. An aerobe is a microorganism that lives and grows in the presence of free oxygen. The microaerophile is an aerobic bacterium that requires oxygen at a concentration less than that in the atmosphere. And the anaerobe is a microorganism that grows best or excessively in the absence of oxygen. Another thing that can determine growth of microbes is relationships. We've got 4 different types of relationships. Parasitism. It's a relationship between 2 organisms in which the host is harmed in some way while the colonizer benefits. So one benefits and one is harmed. Commensalism is an unequal relationship in which one species derives benefit without harming the other. So they live in a pretty happy zone. Antagonism is a relationship in which microorganisms compete for survival in a common environment by taking actions that inhibit or destroy another organism. So these are not happy. And the synergism is the coordinated or correlated action by 2 or more drugs or microbes that result in an heightened response or greater activity. So these are several different types of relationships. Some are happy, some are not. Some get hurt, some don't hurt others. They live in a happy place. These can all affect microbes and their growth. One concept that affects growth and population is something called binary fission. And it's the formation of 2 new cells of approximately equal size as a result of a parent cell division. So you have the parent cell, and then you have the replication by DNA, and the segregation, and then the cell splitting into 2 different parts. So then you end up with 2 individual cells and not 1. So I actually have an example here that breaks it down on the left-hand side. Another aspect that we have to take into consideration with growth is generation time and the growth curve. Generation time is the required time for complete fission cycle, and this is from parent cell to 2 new daughter cells, also called doubling time. The next one is the growth curve, and it's a geographical representation of the change in population size over time. So you've got a graph, and then it'll show you like you have 500, and then you'll have the little lines that go up or down or whatever. It'll demonstrate population size. And this graph has 4 periods known as the lag phase, the exponential or log phase, the stationary phase, and the death phase. Okay, we're going to talk about our phases really quickly. The first one down here on this chart at the very bottom, I'm going to try to see if we can highlight that. This one right here is the lag phase. It's the early phase of population where growth during which no signs or growth occur. The next one is the log phase, and it's the maximum rate of cells division during which growth is geometric in its rate of increase. Also an exceptional growth phase. So this is this one. Our next one is our stationary growth phase and survival mode in which cells either stop growing or grow at a very slow rate. And that is this one. Not much change between these 2. And then our last one, of course, which is this one, is going to be the death phase, and it's the end of cell growth due to lack of nutrition duplication of environment, and accumulation of waste. Population of cells begin to die.